Right now, inside your gut, there are bacteria that can destroy cancer cells. Not metaphorically, not speculatively. In January 2026, 3 studies were published simultaneously in Nature Medicine, one of the most prestigious medical journals on the planet. The results stopped oncologists cold. Patients with lung cancer, kidney cancer, and melanoma who received transplants of healthy gut bacteria saw their response rates to cancer treatment nearly double. In the lung and melanoma trial, 75% of patients who received the gut bacteria transfer responded to treatment, compared to 50 to 58% in patients who didn't. That's not a marginal improvement. That's the difference between treatment working and treatment failing. And here's the part that should make you stop what you're doing. You don't need a transplant to protect that system. You just need to stop starving the bacteria you already have. Because right now, today, the modern American diet is systematically destroying the microbial army that stands between you and cancer, heart disease, and chronic inflammation. And almost no one is talking about it in terms this direct. Here's what you were never taught about your digestive system. Your gut is not just a tube that processes food. It is an immune organ. It is a hormone factory. It is a living ecosystem of roughly 38 trillion microorganisms, bacteria, fungi, viruses, that produce compounds your body cannot make on its own. Vitamins, neurotransmitters, anti-inflammatory molecules, and something that's now being called the most important discovery in metabolic medicine in a generation, natural GLP-1. You've heard of Ozempic. You've heard of Wegovy. Those drugs work by mimicking a hormone called GLP-1, glucagon-like peptide 1, that signals your brain to stop eating and regulates blood sugar. What the pharmaceutical industry doesn't lead with is this. Your gut already makes GLP-1 for free, without needles, without side effects. When you eat fiber, specific gut bacteria ferment it and produce a short-chain fatty acid called butyrate. Butyrate directly fuels the L cells in your intestinal lining, the same cells that produce and release GLP-1 naturally. That GLP-1 travels to the brain and shuts off hunger for hours. The bacteria that do this are living inside you right now. The question is whether you're feeding them or killing them. The average American eats 10 to 15 grams of fiber per day. The recommended minimum is 25 to 38 grams. Most research on gut microbiome health points to 30 to 50 grams as optimal. That gap between what Americans eat and what the bacteria need is not a small oversight. It's a slow extinction event happening inside your digestive tract. Here's a simple test. Look at your plate from yesterday. Was it mostly beige, white bread, pasta, rice, chicken breast, crackers, chips, maybe a handful of iceberg lettuce that barely counts? That plate is sterile territory for the bacteria that protect you. There's nothing for them to ferment, nothing to produce butyrate from. The GLP-1 signal never fires. The hunger never fully shuts off. The immune surveillance weakens, and it compounds over time. Studies on gut microbiome diversity show that when specific bacterial species go unfed for extended periods, their populations collapse, and they don't always recover when you start eating well again. The damage is cumulative. Now, here's the mechanism that connects this directly to heart disease and cancer. When the protective bacteria die off, the gut lining weakens. The tight junctions between intestinal cells, the barrier that's supposed to keep bacteria and toxins inside the gut, begin to fail. Researchers call this increased intestinal permeability. Most people know it as leaky gut. When that barrier breaks down, bacterial toxins called lipopolysaccharides leak into the bloodstream. Your immune system detects them as a foreign invasion and fires an inflammatory response every day, chronically, for years. That chronic low-grade inflammation is now understood to be a primary driver of atherosclerosis, the hardening and narrowing of arteries that leads to heart attack and stroke. Research published in Cardiovascular Research and in Circulation Research has identified the gut-heart axis as one of the most important and least discussed mechanisms in cardiovascular disease. The same inflammation promotes the cellular environment in which cancer takes hold and spreads. 1. Depleted microbiome. 2 of the leading killers in the United States. Let's go back to those 3 studies, because the details matter. The first trial, called FMT Luminate, enrolled patients with non-small cell lung cancer and melanoma, 2 cancers with historically variable responses to immunotherapy. Participants received capsules containing healthy donor gut bacteria before starting their cancer treatment. The results: 75% response rate in melanoma patients compared to 50 to 58% in the standard treatment group. Encouraging enough that researchers are now designing larger trials. The second trial, called Perform, tested the same approach in metastatic kidney cancer, a cancer where immunotherapy side effects are often severe enough to stop treatment entirely. The result? The FMT not only improved response, but also significantly reduced the toxic side effects of the immunotherapy drugs themselves. The third trial, also in kidney cancer, was a randomized, placebo-controlled Phase 2 study, the gold standard of clinical evidence. Patients who received gut bacteria transplants from complete immunotherapy responders showed significantly improved progression-free survival compared to the placebo group. The proposed mechanism is not mysterious. Healthy gut bacteria produce compounds that activate T cells, the immune cells that identify and attack tumor cells. When the microbiome is diverse and well-fed, T cell activation is robust. When the microbiome is depleted and inflamed, T cell activity is blunted. The cancer has a quieter immune environment to grow in. Researchers from the University of Florida published a related finding in late 2025. A small compound produced naturally by gut bacteria more than doubled the response to lung cancer immunotherapy in animal models, and has now been synthesized into a drug candidate for human trials. The science is not fringe. It is nature medicine. It is peer-reviewed. It is reproducible. And it points in one direction. The gut microbiome is not a side topic in cancer medicine. It is becoming a central one. The villain in this story is not one dramatic thing. It's the accumulated weight of ordinary American life. Ultra-processed foods. By now, most people understand these are not ideal, but the specific mechanism matters. Ultra-processed foods don't just fail to feed good bacteria. The additives in them, particularly emulsifiers like carboxymethylcellulose and polysorbate 80, directly damage the gut lining and suppress microbial diversity. Multiple studies have shown that common food emulsifiers disrupt the mucus layer that protects gut bacteria, causing the very leaky gut discussed earlier. Antibiotics. The United States prescribes antibiotics at one of the highest rates in the developed world. Every course of antibiotics is a broad-spectrum strike on the gut ecosystem, killing beneficial bacteria along with the target pathogens. Studies show it can take 6 months to 2 years for the microbiome to partially recover, and some species may never return to pre-antibiotic levels. Chronic stress. Cortisol directly suppresses beneficial lactobacillus and bifidobacterium populations while promoting the growth of inflammatory pathogenic species. Americans are the most stressed developed world population by most self-reported measures. The gut ecosystem reflects it. Low dietary diversity. Not just low fiber, low variety. The specific bacterial species in your gut are like specialists. Each one ferments slightly different compounds, responds to slightly different foods. A microbiome with 50 species is fundamentally more resilient than one with 10. Research from the Human Gut Project consistently shows that people who eat 30 or more different plant foods per week have dramatically higher microbial diversity than those who eat fewer than 10. Here is what the research actually supports. Not as a supplement protocol, not as a detox, but as the foundational dietary shift that feeds the bacteria you already have. Step 1. Add color before you subtract anything. The most durable change is additive. Don't start by removing foods. Start by adding plants of different colors to every meal. Each color represents a different class of phytonutrient that feeds a different bacterial population. Red, orange, yellow, green, purple. Each one matters. Aim for 5 different plant colors per day as a starting point. Step 2. Introduce a fermented food daily. Real fermented foods, plain yogurt with live cultures, kefir, sauerkraut, kimchi, miso, introduce living bacterial strains directly. A landmark study published in Cell in 2021 found that 10 weeks of high fermented food intake increased microbial diversity and reduced 19 markers of systemic inflammation. Not a supplement trial. Real food. 19 inflammation markers. Step 3. Feed. Don't just populate. Probiotics without prebiotics are like planting seeds in concrete. Prebiotic foods, garlic, onions, leeks, asparagus, oats, green bananas, legumes, are what your existing bacteria eat to produce butyrate and GLP-1. The bacteria you already have need to be fed before anything else. Step 4. Protect the barrier. Reduce ultra-processed food with emulsifiers specifically, not because of calories or fat or sugar, but because emulsifiers mechanically disrupt the gut lining. Read ingredient labels for carboxymethylcellulose, polysorbate 80, and carrageenan. These are in salad dressings, ice cream, processed meats, and most packaged breads. Step 5. Be consistent for 3 weeks minimum. The gut microbiome responds to diet within 24 to 48 hours. But structural changes, increases in beneficial species populations, improvements in gut barrier integrity, take 2 to 3 weeks of consistent feeding. It's not a fast. It's not a cleanse. It's a sustained shift. Here is what I want you to hold on to. 3 clinical trials in Nature Medicine. January 2026. The gut bacteria inside cancer patients, when properly supported, nearly doubled treatment response rates. That finding didn't come from nowhere. It came from decades of research showing that the 38 trillion organisms living in your gut are not passive passengers. They are active participants in your immune function, your hormonal regulation, your cardiovascular health, and apparently your capacity to fight cancer. The research is there. The mechanism is understood. The dietary changes that feed this system are not exotic. They are not expensive. A cup of plain yogurt. An extra vegetable with dinner. Garlic in the pan instead of garlic powder in a packet. What is not there? In most doctors' offices, in most nutrition guidelines, in most conversations about cancer prevention, is the urgency this evidence deserves. That's what this is for.